I'm going to go over some of the information regarding DNA and just review a little bit of the structure and the important things that you need to know before we get into how DNA is used in the lab. So please make sure you're taking Cornell notes on the information. They will be collected and they will also be useful for your first quiz coming up. All right, so let's start with a review of the basics of the structure of DNA. So as we know, DNA stands for deoxyribonucleic acid, and it's a molecule that encodes the genetic instructions used in the development and functioning of all known living organisms, and also some viruses. DNA is a nucleic acid. So we talked about 4 macromolecule types in biology, nucleic acids being one of them. And along with proteins and carbohydrates, nucleic acids create all of the molecules that are essential for all life forms. So the question at the bottom asks, why do you think DNA molecules consist of 2 strands? And this is something that we've also talked about. But the primary function of DNA is to store and transmit genetic information. And so it's a really important molecule. In order to accomplish that function, DNA has to have 2 properties. It has to be stable, and we have to reduce the possibility of damage to the molecule because it is so important, because it encodes all of your genetic information. So DNA also has to be capable of copying itself. And so the 2-stranded structure of DNA gives it both of those properties, stability and also the ability to copy itself. So we have essentially created a template for the molecule. So one side tells us what goes on the other side of the molecule. If something were to happen to one side of the DNA molecule, we could go back and fix it, knowing the bases on the corresponding side. So most DNA molecules consist of 2 strands coiled around each other to form a double helix. So that's the shape we saw on the last slide. The 2 DNA strands are known as polynucleotides since they're composed of smaller units called nucleotides. Remember, we talked about monomers and polymers when we talked about it in bio. So polymer is the really, really long chain of repeating units. So each little nucleotide attached together, and then the monomer being each nucleotide itself, or the building blocks. Each nucleotide is composed of a nitrogen-containing base. So these are the letters that we talked about. So G for guanine, A for adenine, thymine for T, and C means cytosine, as well as a monosaccharide sugar called deoxyribose, so hence the name deoxyribonucleic acid, and a phosphate group. The nucleotides are joined to one another in a chain by covalent bonds between the sugar of one nucleotide and the phosphate of the next. Remember, we drew this on that template in bio. You had it folded and you created one strand of your DNA molecule with the little sugar house and the phosphate group and the base pointing towards the middle. So this here, this yellow piece is the now I just lost my train of thought, is the nucleotide. And so here's the sugar house, and then this is the phosphate group. So we usually draw this as a circle, sugar house, and then here's our base, A, T, C, or G, pointing towards the middle. And so the bases are facing each other. So the nucleotides are joined to one another in a chain by covalent bonds between the sugar of one nucleotide and the phosphate of the next. So this is the covalent bond here binding the sugar house. This one right here, the sugar house and the phosphate group. And so we have that sugar phosphate backbone. According to base pairing rules, we know that A is always going to bind with T and C is going to bind with G. And so that helps with the stability of the structure, and it also helps with if something goes wrong with one side of the molecule, we're able to repair that using the other side as the template. Hydrogen bonds are going to hold these 2 guys together and hold both sides of the molecule together and make it double-stranded. So this should also be a review. But Francis Crick and James Watson were really essential in understanding the structure of the DNA molecule. Crick was a molecular biologist, and played a crucial role in research related to revealing the genetic code. He's widely known for the use of the term central dogma and having solved the structure of DNA with James Watson in the 50s. The central dogma is illustrated above, so this is also something that we focused on quite a bit in biology. It has also been described as DNA makes RNA makes protein. However, that statement's a little bit oversimplified. Crick's central dogma idea does not say that information cannot flow in the reverse from RNA to DNA. It does rule out the flow from protein to RNA or DNA. In other words, this big idea, or this really important idea in biology called the central dogma, states that such information cannot be transferred back from protein to either protein or nucleic acid. So typically we have our DNA strand, then we open that DNA strand up during transcription, make a copy of it, it gets translated into our RNA molecule, and then that RNA is then made into the protein that is so important for creating the structure of our bodies. So here's kind of this idea in better detail. So transcription of DNA to RNA to protein. This is the dogma that forms the backbone of molecular biology, and it's represented by 4 big stages. So in step number 1, DNA replicates its information in a process that involves many enzymes, so replication. Then the DNA codes for the production of messenger RNA during transcription. So transcription is when we're actually reading the DNA code and we're creating that mRNA copy, the messenger RNA, which will then move out of the nucleus, which is step 3. In eukaryotic cells, the mRNA is processed and migrates from the nucleus to the cytoplasm. So remember, our DNA is stuck inside of the nucleus. We want to make sure that it's contained and that it's safe, and then it's not going to be harmed by the enzymes that are in other parts of our cell. So we have to keep it safe, but we also need a way to get the DNA code out so that we can make proteins at the ribosome. And the way we do that is through the messenger RNA molecule that does travel from the nucleus into the cytoplasm to the ribosome. And then the final step, the messenger RNA carries the coded information to the ribosome. The ribosomes read the information and use it for protein synthesis. And when we're reading it, we're reading this code and essentially translating it into amino acids and the order that the amino acids go in. So that's why it's called translation. Proteins do not code for the production of protein, RNA, or DNA, but they are involved in almost all biological activities, structural or enzymatic. Proteins are exceptionally important for our bodies, and it's essentially proteins that are doing all of the important things, but the code for that protein again comes from DNA that is in the nucleus. All right, so hopefully this was all review. If you want to review some of the videos that are on YouTube, you can do that. Just you need to know the basic structure. So it's double-sided, it's a double helix, A's, T's, C's, and G's. And then the way that we do, in fact, make protein through the central dogma. So DNA makes RNA, makes protein through replication, transcription, and translation. All right, thanks, guys.